In this module, we will continue to study the importance of the second messenger, which is required for the cell signaling. Now, some of the hormones that achieve their effect through cyclic AMP, as we studied previously, as a second messenger, they are adrenaline, glucagon, and luteinizing hormone, which are important second messenger carrier. Now, the function of cyclic AMP is that it is used for intracellular signal transduction because it diffuses into the cell very easily. The second is its regulation of the protein kinase. It regulates the protein kinase by cyclic AMP, which is the phosphorylation of protein kinase, and the regulation of cyclic AMP inducible gene expression via protein kinase, that is, it induces a gene expression through the protein kinase. Just as in this figure, the cyclic AMP, it is going to the protein kinase, then cyclic AMP again, it is further processed, making the signal which is diffusing into the nucleus, and via ADP, it is further going to change the cytoplasm, to change the nucleus signals, and to change the transcription for the DNA. Now, phospholipids of the cell membrane, they are very, very important. And phospholipids, they are converted into the also second messenger. And they are converted by a variety of enzymes. Different enzymes are there which convert them into the second messenger, and they, as a matter of fact, act as a regulatory response in the extracellular signals. जो सेल के बाहर के सिग्नल हैं उनको सेल लाइपर्स जो हैं वो उनको ट्रांसलेट करते हैं. फॉर एग्जाम्पल यू कैन सी देयर आर फैटी एसिड्स and a structure of generalized phospholipids is fatty acid, it is phosphorylated, and then it is going to make a change. Now, these enzymes, they are phospholipases, that is the lipid splitting enzymes, phospholipid kinases, which are lipid phosphorylating enzyme, and phosphatases, which are lipid dephosphorylating enzyme. That is, these are the enzymes which deal or which treat the lipid as a substrate and convert them into the different second enzymes. Now, here, the generation of a second messenger is as a result of ligand-induced breakdown of phosphoinositide PI in the lipid layer of the membrane has been demonstrated that how in the channel the lipids, they are being converted to the different enzymes and to make the change in the cell to carry out the signal in the cells. Now, different responses which are mediated by the protein kinases are like these are tabulated here. Some of them are in the blood platelet, that is the serotonin release. Similarly, adrenaline medulla, that is the secretion of epinephrine, and so on. In the thyroid, secretion of is calcitonin, testes, testosterone hormones, and so on and so forth. There are different responses which are mediated by the protein kinase G. Now, protein kinase G, as we have seen earlier, has a number of important roles. One is cellular growth, and differentiation. Then there is a cellular metabolism, cell death, transcription activation, the gene formation, and the DNA activity. Now, calcium, which is also the second messenger we studied, is very important. And calcium ions, they bound different target molecules and they trigger the specific responses. An earlier experiment that was conducted in which single liver cell was injected with acurin. Acurin is a protein of a jellyfish that luminesces when it binds to the calcium ion, and that luminous mean जब ये चमकता है, ग्लो करता है, तो इससे वो जो इसकी ल्यूमिनेंस है, वो द इंटेंसिटी ऑफ ल्यूमिनेंस इज प्रोपोर्शनल. जितना कैल्शियम होगा फ्री उतनी ज़्यादा ये ग्लो करेगा, इज अ प्रोपोर्शनल टू द कंसंट्रेशन ऑफ फ्री calcium ions. Now, exposure of the cell, another vasopressin, it leads to controlled spike in the concentration of free calcium at periodic intervals. दूसरा इसका ये सबूत भी है. So calcium is also very important molecule as a second messenger for the cell signaling.